Good afternoon, everyone. Microphones on. Good. We're going to wrap up this conference, which has been great. And as far as I recall, it's been one of the rare with 2 sessions. So I'm sure you've been moving around to get as many talks as you could. Thanks to our technician, Amanda. We're going to get all of them on YouTube. And sometimes you'll be able to catch up on what you didn't see. But for the one that chose to be here this afternoon, we're going to study and listen to population monitoring. We have 5 talks. I'm going to keep everyone online so we can finish. Some people have a train or a plane to catch. So let's try to wrap it up on time. Our first speaker is Issac Acker. He's going to talk about fungal disease. I'll be sitting right here with my timer and give you a 12 minutes note. That's good for you?
Yep.
Let's go.
All right. Good day, everyone. My name is Issac Acker, member of the Riley Lab at Mount Allison University, and I'm here to talk to you about testing for the presence of Ophidiomyces field in New Brunswick and Nova Scotia. Now, recently, wildlife disease has become a major threat for reptiles and amphibians worldwide. Diseases such as mycoplasma and chytrid fungus have caused widespread decline in the respective host species. These diseases are often allowed to spread unnoticed due to the lack of research around the persistence and prevalence in their host areas. Now, this is currently occurring with the disease right now in Canada, and its name is Ophidiomyces ophiodiicola. Now, what is Ophidiomyces ophiodiicola? Well, this is a recently discovered fungus which lives in the soil, and its only known host is snakes. This fungus can cause an infection in snakes called a snake fungal disease infection once it gets past the snake's epidermis after the snake comes into contact with either infected soil or other infected snakes or snake skins. Once inside the snake, this snake fungal disease can cause symptoms like lesions, and once it enters through the tissue, can cause potential mortality in individual snakes. Now, currently, Ophidiomyces ophiodiicola is known to occur worldwide in captive populations, varying all the way from Australia to Japan. It has also been found in free-ranging populations of snakes in both North America and Europe. Within North America, Ophidiomyces ophiodiicola has been found across the United States, and in Canada, testing has shown that it is widespread in Ontario and it is potentially endemic to the area. However, in the rest of Canada, there has been little to no research done on the presence of this fungus, even though there is potential that this fungus could be endemic to the area or have novelly spread from Ontario or the United States. This leads to my research question of, is Ophidiomyces ophiodiicola present in New Brunswick and Nova Scotia? In order to answer this question, I did a study which revolved around sampling for free-ranging and preserved specimens from May of 2022 all the way through to October. During this research, we would look for this fungus in both of these 2 provinces, while also sampling any live, deceased, snake skins, or any preserved specimens that we had found in the field. When we were looking for these snakes, we wanted to make sure to survey a variety of different habitats for these individuals. This is due to the case that Ophidiomyces ophiodiicola could exist in any of these habitats, including high-risk habitats such as international ports or just international boundaries between Canada and the USA. We were lucky enough to be able to survey a total of 46 different sample sites for these snakes during our survey. Now, for our methodology revolved around capturing free-ranging snakes and swabbing them with a cotton swab on both the ventral and dorsal side of the snake. We would also check these snakes for signs of snake fungal disease lesions, and if we observed any potential snake fungal disease lesions, we would swab them with an additional cotton swab to see if we can determine if these lesions are indeed caused by snake fungal disease. We'd also collect any carcasses of snakes or snake skins that we would find in the field and swab those as well. We would then take GPS locations of the individuals that we had found in order to map the distribution of our samples and the potential distribution of the fungus if we did find it there. We also took general photos of our snakes and lesions for future reference. Now, we were lucky enough to be able to sample individuals at several institutions throughout the Maritime provinces. One of these was the Mersey-Tobec Research Institute in southern Nova Scotia, and the other was the Museum of New Brunswick in western New Brunswick. These institutions had snakes they had collected dating all the way back to 2010, so it gave us a real good notion of the potential of snake fungal disease existing in our native snake populations starting from 2010 all the way up through to 2022. The surveying for these individuals were relatively similar, along with sampling for snake fungal disease, as we would follow the same swabbing methods. But we would also take small tissue samples from these snakes, as seen with this northern ringneck snake, in order to determine if there was indeed snake fungal disease present in the tissue if we found Ophidiomyces ophiodiicola present on the skin of the snake. After we finished collecting all of our swabs, we would then send off our body swabs, our lesion swabs, and our snake skins off for qPCR analysis to the animal health laboratories at the University of Guelph, which has been used to determine if Ophidiomyces field has been present on snakes in previous studies in Ontario. Now, as for our results, we were able to survey a total of 158 different snakes and snake skins in both New Brunswick and Nova Scotia, with the majority of these snakes being maritime garter snakes, and there being one lucky common water snake that was in the collection of the Museum of New Brunswick, which was not in our survey area, but we swabbed it anyways. Now, this map here helps to show the distribution of all the snakes we sampled throughout our research, with the diamonds representing snakes that were sampled alive, squares representing snakes that were swabbed dead, and the apostrophes showing snakes that were swabbed just outside of our study area in Maine. Now, as you can see, some of our species, like the maritime garter snake, had a wide distribution where we were able to sample them, while other species, like the northern ringneck snake, we can only find in Nova Scotia. We also did not include any of our snakes that we surveyed that were the eastern ribbon snake, as they are currently a data-sensitive species. Now, while we were surveying for these snakes, we had observed several potential snake fungal disease lesions on these snakes, with most of them being present in maritime garter snakes. However, until we receive our qPCR results, we cannot determine if they are indeed snake fungal disease lesions. This is because in the snake's environment, there are several other factors that could cause these potential lesions, such as predation attempts, other pathogens, or just general wear and tear on the snake as it moves through its environment. Now, for our genetic findings, the qPCR results show that there was no positive cases of Ophidiomyces field in any of our samples. But more interestingly, there was not even a trace of any Ophidiomyces field DNA found at all in any of our snake skins, body swabs, or the lesion swabs. This meant that we did not have to test any of our tissue samples, as we couldn't even find a trace of this fungus on our snake's epidermises. So, what does this tell for us about Ophidiomyces field in Canada? Well, it shows that even if it is indeed novel or endemic to the Ontario or United States areas, there is something that is preventing it from being able to exist in our native snake populations of New Brunswick and Nova Scotia. Now, it is possible that there might be a barrier that is limiting these snake populations from transferring this fungus to our native snakes. One of the potential barriers to why we're not seeing this disease in New Brunswick and Nova Scotia is host susceptibility. Now, Ophidiomyces field does not affect all snakes the same. As shown by former research in central Canada, it has shown that some species, like the garter snakes, are potentially more resistant to Ophidiomyces field infection compared to other species. Luckily, for New Brunswick and Nova Scotia, these maritime garter snakes are the most prevalent snake species we have around. They are our ditch snake. You can always find them. So, this might mean that even if Ophidiomyces field were to arrive in New Brunswick or Nova Scotia, it would not be able to find enough vectors or susceptible snake species, like this cute little northern red belly snake, in order to be able to persist and spread throughout the province. Now, let's compare this to a place where we know Ophidiomyces field to occur in, central Canada. There exists a wide diversity of snakes here, like the eastern fox snake and northern water snake, which are known vectors and are easily susceptible to this disease. This might be indeed why we're not seeing Ophidiomyces field here. We might just not have enough snakes that are susceptible enough to maintain a population of this fungus. Now, there is also several other potential reasons to why we're not seeing this fungus here, which associate with barriers, such as the Great Appalachian Mountain Chain limiting snake spread eastward, to the maritime provinces just generally having a smaller population and not as many captive herps, which might limit the ability of Ophidiomyces field to spread from our captive to native populations, all the way to just general climate limiting the spread of Ophidiomyces field northward from the United States. Now, what does this mean for our future direction for this research? Well, it means that we need to continue to survey for Ophidiomyces field across all of Canada in order to determine its real persistence in the area. We also need to create a national monitoring program like those used for SNAPs for BSAL in order to be able to pull up all our data in order to really answer this question of what Ophidiomyces field is doing in Canada and what snake populations is it actually present in. Finally, we need to continue to prioritize testing for this fungus, not only just in general, but at the high-risk entry points where this fungus could be introduced into Canada, such as from areas like the States, or through the captive pet trade, which is why we need to test our captive populations in order to gauge the risk, not only for our native species, but for some of our captive species that we use for head starting. Now, why should you care about this research? Well, This is the first ever survey done for Ophidiomyces filticola ever in New Brunswick and Nova Scotia. Not only that, but it showed that there was no presence of any Ophidiomyces filticola found at all in any of our samples, which is different compared to those tests in Ontario, which found it to be quite prevalent across the province. Not only does this mean that potentially our snakes have not come into contact with this fungus, but they also might be potentially vulnerable if this fungus were to arrive here. Certain species of snake, like the eastern ribbon snake, have a generally small population and are quite isolated, and if this fungus were to arrive here, it might negatively impact their ability to recover and persist within the province. Now, I would like to thank my volunteers, lab mates, and landowners who helped me with this research, along with Dr. Graham Forbes. I would also like to thank Laurie Finney from the Mesozoic Research Institute and Don McAlpine from the New Brunswick Museum, who provided me with so many samples. I would also like to thank my supervisor, Dr. Julia Riley, and mentor, Dr. Baxter Gilbert in the back, who really made this research possible.
So to find disease, you need to find individuals. So our next talk is going to talk about capture probability. I think it's a work done in Canada, but our speaker is from New Zealand. So a lot of travel to work here. Dr. Armstrong.
Thank you, David. I'm going to have to start by correcting that. Now, I'm from Canada. I can use that one. Okay, I'll use that one. It's for short people, though. That's fine. I'm from Canada, but I do live in New Zealand most of the time these days. So thank you for bringing it up. Start by acknowledging for this talk. On the screen there, the person who did all the work, or most of the work, is Patrick Moldovan, who's the first author there. Patrick just completed a PhD quite recently with the Spotted Salamanders at Algonquin Park. Glenn and Niall there were the supervisors, and at the moment, and for the present talk, I'm basically the groupie. And that didn't work. I'll try that. Oh, there you go. I just had to hit it harder. Okay. And I just wanted to give a talk about salamanders, especially mole salamanders. I've been obsessed with mole salamanders since I was a kid. And the reason I was obsessed is because we used to go up to the cottage. And guess when? In July and August. And I would look and look and look for mole salamanders and not find any. And of course, when I got older, I started to learn about their life cycle and that all the action takes place early in the season. There's photos over there. You can see that a salamander emerging just at the snowmelt there, rushing off to the nearest vernal pool. So that pool there is one of our vernal pools up at our cottage. Then you get these eager males waiting in the pools for the females turn up, and then it's all on. And, by May, or even by early May, all you see is egg masses. And even they then disappear because, of course, the larvae are then so small, so then you got to wait for a month or two. And then finally you see these larvae. That's really cool. And then they're kicking around. And then just recently, in the last month or so, the metamorphs come up. So they've got this wonderful life cycle once you kick into it. Now, today I'm going to talk about Algonquin Park. And I came to Algonquin Park as to work in the turtle mines first in 1981. So most of my association has been with the Snapping Turtle Project, collaborating with Matt and others. But to go to the research station, you basically go right by Bat Lake, and you have to find out about Bat Lake. So Bat Lake is 3.4 hectares, and it's a kettle lake, which means it's been gouged out by a glacier. It has no inflow or outflow. It has no fish. It's amphibian paradise. And getting there and saying, oh, and there's this lake over there, and it's probably full of thousands of salamanders. And I'm, oh my God, that's so cool. And then years later, really starting full on about 2008, a long-term project started up with the spotted salamanders, which has been what Patrick has picked up on. I don't have time here to really talk about, the bigger, biological and management questions and so forth with this project. I'm just talking about abundance estimation here. I think we probably all agree that monitoring can be useful and that monitoring abundance can be useful. Just monitoring trends in abundance can be useful for conservation, but if you can do it, it's even better to be able to actually estimate abundance. And many people have talked about that. And many people here have also talked about capture mark recapture, which in the absence of being able to do a direct count is generally our our gold standard. So my little matrix at the bottom there is just showing the typical encounter history, which many of you have worked with, where you've got individuals, animals as rows, and the ones that show the fact different surveys where you've encountered an animal or not. There's the zero showing when you haven't. Okay, so what do you need to do mark recapture? Well, you don't actually necessarily need to mark or recapture, but you do need to do some sort of repeated surveys, and you do need to be able to identify your animals. In the case of spotted salamanders, handily, they have all these little spots, and which, if you spend enough time on it and have some fancy software, they're individually identifiable. Just to either remind you, or for those who may be less familiar with mark recapture, there's sort of three categories. There's your closed mark recapture, where you just want to do a snapshot estimate of abundance. You just go out and survey in rapid succession. There's your open mark recapture, where you're doing it over a longer term to get survival and recruitment as well. And then there's the mixture of the robust models, which some people have actually talked about today. Algonquin Park. Yes, Stephen. Yes, just at the back there. It kindly took me out with Taylor there shown in the picture in 2016. So I got to see this in action. So there's that lake on the left there. And those little dots you can see around here are all minnow traps, and they're just about 8 to 10 meters apart across the around the whole lake. So it's breeding population surveying of animals that have come into the lake to breed. And you can see Taylor there is holding a minnow trap, and he's very happy because it's full of salamanders. So basically, they all get pulled out, and the people like Taylor and Stephen who are doing all the hard work are then processing the animals. There again. Okay, so I'm just going to switch now to the talking about what Patrick found in his thesis. Patrick did a lot of thinking about how he was going to deal with the data, and he wanted to estimate abundance, he wanted to estimate survival, and to be able to do this over time and to pick up trends. Okay, and so over it's 12 years, 2008 to 2019. There were 2,143 individual males and 939 individual females were captured. Patrick decided to analyze it with a robust model, and what he ended up doing was sort of treating each 3 days survey as a survey. Okay, and typically it's about a month from ice melt, basically until the salamanders stop coming into the lake. So then you're getting about 10 different surveys over each of those 12 years. Okay. He found with the females that they had too few recaptures to actually be estimable, but with the males, he got some estimates. Okay, so look, lots of estimates there over the years, and I can't actually read this, and I can't read that, but you see it's in the hundreds. And so, okay, that looks kind of plausible. Right? It kind of jumps up and down kind of suspiciously, and the confidence intervals are suspiciously small, but besides that, it looks plausible. Okay, so what did Patrick do? Was he happy with that? No. If you know Patrick, you know he keeps going. So, and this was, I guess, about 2016 was last time I was there in the summer until the current year. Patrick had this mad idea that they're going to put a drift fence around the whole lake. So that and various other people went and put this drift fence, which you can see my photo there. It's just a little metal fence around that long with various cover objects, and that went around the whole lake. And then cover objects and traps, and of course, then you have teams of people that go out every night and then every morning, again through this whole period, and basically count every salamander coming into the lake to breed. Every wood frog, everything else they find. So it was amazing, a huge amount of work though. And guess what happens? Okay, so the blue on the graph is the same as was before. what I've added there is those horizontal lines there in blue for the boys, pink for the girls. Okay, you see, despite there being a lot fewer females caught and very few recaptures, there are actually more females than males coming into the lake. And the other thing is, if you look at the years where there's overlap here, it's just these 2 years here, you can see here's Patrick estimates from the robots model, and these are the number of males they counted. So there's about 4 times as many individuals being counted coming in as were being picked up. So why is this? So that's really what I guess how I got involved in this year, and we thought, well, why would this likely to be? Well, for those of you who know mark-recapture, one of the really obvious reasons is you've got individual heterogeneity or individual variation. Some animals tend to be captured and some don't. It could be personalities, it could be where the traps are with respect to the animal's habits. It could be a whole lot of reasons. If you look again carefully at my mark-recapture history there and imagine that those are 6 different individuals that were there during all of those, whatever, it's about 10 surveys, you'd then say, that's an example of pretty clear heterogeneity. The person at the top obviously loves to be captured. He gets captured every time. And then as you go down, you see in the last 3 just never get captured at all, even though they're there. So how do you actually deal with that? Well, we've got some reasonably good methods now, and the best methods are using Bayesian hierarchical modeling, which basically means you fit a normal distribution to the variation you see in the capture probability. Okay, and that's really easy if all you want to do is survival. If you want to do abundance, it creates problems because it's the individuals that you've never captured. They're the interesting ones. You know how many are there that you captured, but you don't know how many are there that you haven't captured. And how do you measure individual variation in something you never see? So in 2007, Andy Royal and colleagues came up with a method. This was the first published example I know, called data augmentation. And what you do, and you can see at the top 3 there, those are observed animals. So there'll be your encounter history. And what you're imagining is there's a whole bunch of animals out there that you've never observed. Okay. And the idea of this technique is you basically just add this huge number of rows of zeros to your data. And what the 2 categories is there is that the first 3 are unobserved ones that were there hypothetically, and then below that are ones that didn't actually exist. So part of the modeling process is actually modeling how many of these rows of zeros actually represent animals that exist versus ones that didn't exist. And the cool thing, because you've got these rows of zeros, you can then treat them as individuals and model their individual characteristics, including their detection probability. So it's a really neat technique. And to me, as a field biologist, I find it intuitively intuitive. The model fitting is just absolute magic, but it is intuitive. Okay, so for those of you that are familiar with this sort of thing, I was using the CMSA formulation of the Jolly-Sieber model to deal with this, again, with data augmentation. And as I mentioned, individual random effects on detection probability. So you've got this individual variation detection probability, and I also modeled the variation over time as a random effect as well, and basically set it up in a way that the females could kind of borrow from the males. I won't deal with that too much. Added 12,000 rows of zeros for males and 14,000 for females, which is insane. But lo and behold, if you actually look at the estimates now against how many were caught at the fence, it now lines up basically perfectly. It was magic for the males, and for the females, at least the credible intervals do include the number that they were intercepted. So my time's just about up, so I'll just shorten the summary to really 2 points. One is that this method magically did actually seem to work. So we got decent trends for both males and females. The warning for those of you that are going to rush off and do it is if you're adding this many individuals to your data set on my computer, it took a week to run, and then it crashed. Right? And I haven't subjected my computer to it as well. So I was using a program called OpenBugs. For those of you that are Bayesian modelers, I think you need to try using Stan for this, because that's probably a lot faster. Okay, acknowledgements. Zillions and zillions of people who've worked on this project did all the hard work. And then, of course, the station, always a wonderful place, always very warm and welcoming, supported this. And then Ontario Parks and Insert for supporting the research. Thank you.
Now we're going to welcome local researchers whose title has changed a little bit. We're still about turtle, and this is Albana.
Okay, thanks everyone. My name is Albana. Sorry about the title mix-up. But today we're going to be talking about understanding long-term population trends of northern map turtles. I just wanted to say thank you to my co-authors for their support and advice throughout this project. I wanted to say thank you to our funders for their support, and also thank you to the community for their time and for their knowledge and for making this research possible. Okay, so local ecological knowledge, also known as LEK or LEK, is this knowledge base that's based off of the ecological relationships that people have with their environment. And this knowledge base is super in-depth and provides a lot of nuance about the environment and about the environment changing over time. So local ecological knowledge has been used, especially recently, as a knowledge base to provide us information about changes to wildlife populations. And it's because of this, we asked the question, what has happened to the northern map turtle population over time? And we use local ecological knowledge to help us understand these changes. The northern map turtle, as we have heard throughout these past couple of days, is a species at risk. It's listed as a special concern federally and provincially. We asked locals also what they predict will happen to the future of northern map turtles based on their knowledge. And we also asked if they have any advice for conservation measures that will work on the lake. Ophidiomyces Lake, as Greg mentioned yesterday, is the lake that Cubes is based off of. So I wonder if I can use my mouse. Oh, no. But Cubes is that little blue dot right in the middle of the map there to the left. Yes, to the left. And Ophidiomyces is about this 8-kilometer stretch, narrow lake. Super beautiful. I'm sure if anybody here is going to the field trip tomorrow, you get to see it in person. And the northern part of the lake is 2 locks. So Ophidiomyces Lake is based off of Rideau Canal. And so these locks provide people to go back and forth, up and down the Rideau Canal. And then in the southern portion of the lake, you see some of the property that Queens actually owns for protection. So some of this property actually has shoreline access to Ophidiomyces Lake. So for our study, we conducted interviews with local community members, and we asked that people have at least 10 years of experience on Ophidiomyces Lake. To recruit participants, we tried pretty much everything under the sun. We had advertisements sent out to local lake associations. We posted on community Facebook groups. We posted posters at Cubes. We posted these little tiny advertisements at a local ice cream shop and a gas station. We also chucked these advertisements to random docks on the lake. So we really tried everything. And when we finished up our interviews, which was just over a week ago, so we're still fine-tuning some of these analyses, we conducted some qualitative analyses, including deductive and inductive thematic analysis. So we ended up interviewing 17 participants, and the years that they've been on the lake really varied between 15 to 64 years, and you can see this range in the graph. People mostly were on the lake between May and October, but the time that they were on the lake varied based off of like if they had careers, maybe they didn't have as much flexibility, but later on when folks were retired, they pretty much lived on the lake, so that was pretty cool. And most people frequented the middle portion of the lake, so where Cubes is, to the southern portion. And that's because a lot of the times, especially in the middle of the summer, between the locks and the northern part of the lake, it's very busy with people moving in and out of the lock system. So they stick to the middle to the southern portion mainly. When we asked people based on their local ecological knowledge what has happened to the northern map turtle population since they first visited the lake until now, the majority of them have said that it's pretty much decreased or stayed the same. If anything, it's right between the decrease to stay the same, because folks who have said decrease said a slight decrease. And when we look at the answers a little bit closer, we see that this is all based off of people seeing the turtles bask, and they've been basking in these really large groups and like 40 to 50 and like these large clusters since people have first visited the lake. So that's a good sign. There's no drastic decrease. But folks have also said they've become more aware of the threats that turtles are facing, and they've also noticed less and less turtles nesting on their property, where before they were nesting more. So that's slightly concerning. And I also wanted to mention that participants were really aware of any potential biases and limitations they had in answering these questions, especially based on if they were on the lake. If they were on the lake a little bit more often recently than they were before. So one participant mentioned that ever since they retired, they've been coming in September, and that's usually around the time that turtles are basking more, like the ends of the summer. And even though they said that the population has increased, they said that it could be because now they're coming to the lake at a time where turtles are basking more. And a few other participants mentioned the memory illusion bias, which is when you look back at your memories, they're a bit more exaggerated than what they actually were. So potentially the perception affects the answer that they gave us. And then we asked folks about the future of the northern map turtle population. The majority of them were worried that it's actually going to decrease because of the current threats that the northern map turtle population is facing on Ophidiomyces Lake and the potential future threats that they might face. So looking at some of the threats that they mentioned, the most frequent one was boat traffic and fast speeds and shoreline erosion from wakes. One participant even mentioned that, like, in September, people just want to rip through the gas that they have on their boats, so they're going up and down the lake, especially in the middle area. But the middle area is also the deepest part of the lake, and so a lot of northern map turtles are slowly going towards that and getting ready for hibernation. So this overlap is very concerning. Shoreline development and alterations was another major threat that was mentioned. So this is both private property and folks perhaps not knowing that they can do some of the alterations that they're doing are not great for the shoreline, not just for turtles, but for other wildlife too. But also there's a proposal for a campsite on the lake to expand 4 times what it is currently, and that's going to involve a lot of shoreline alterations for that development. So a lot of the folks that we interviewed mentioned that as a major concern. The use of bubblers and subsidized predators on the lake. So Greg mentioned the other day, we all saw the use of bubblers could increase predation that turtles are facing, but also subsidized predators as the lake gets busier and busier over time. Folks have said that they've seen more raccoons and foxes on their properties. And so this could be a reason why there's been less nestings on people's properties. And another major threat that was mentioned was changes to water quality, and this could include things like plastic pollution from fishing lures or lead pollution from lead weights, but also chemical pollution too. So some folks mentioned that they've noticed a lot of people using products, even like cleaning products, that don't filter well, especially in cottage country, and using septic systems that they don't really monitor or that aren't up to date. And then we also asked people, what are some conservation measures that individuals, but also large organizations could do to help protect the northern map turtle population in Ophidiomyces Lake? So the main one by far was increased awareness and education. And I really love this quote that a participant said. I think most people want to do the right thing. They just need better information on what that actually means. And so people mentioned just joining your local lake association or cottage association to large organizations such as Cubes or other NGOs or other people working in this field to send out pamphlets to cottages, especially as there's been a recent large turnover in cottage ownership on the lake. Enforce and regulate shoreline development. And so some folks mentioned that even though permits are given for people to develop, they don't really read the fine print that they can't develop a certain distance to the shoreline, and they do so anyways. and reducing chemical and plastic pollution and monitoring water quality. So this quote kind of encapsulates those last two conservation measures. I think Ontario has had a very good program with its conservation authorities, and the conservation authorities being watershed based and looking after basically watersheds and water quality and hence shorelines. And so there used to be conservation authorities on Ophidiomyces Lake, but not anymore. And so they were helpful for not just policing, but just spreading awareness as well too, because some people just might not know some of the things they're doing could be harmful to turtles. And then lastly, more protection sites and land trust agreements. So we saw that Cubes owns a lot of that southern land around the lake, but some folks have mentioned just like protecting some of these islands where they've seen a lot of turtles nest could be helpful, especially for recruitment for northern map turtle populations in the future. So why is our study so cool? So this is a case study for local ecological knowledge being used to monitor a long-lived species. As we've seen, a lot of participants in our study have been in this lake for 60 plus years, which is as long as some of the turtles have been on this lake, and that's pretty wild. But local ecological knowledge is also super cost effective. Imagine having to do a field study for 60 plus years. That's almost unheard of. And it also provides a lot of nuance to population trends that sometimes quantitative data alone just we can only estimate. Our study also provides opportunities for cubes and cottage collaborations. So a lot of folks are super interested to learn more. We spent like sometimes half hour to an hour afterwards just talking about the research that we're doing at Carleton, but also at Cubes. It was a pain to transcribe that, but it was just nice to hear that people were interested in what we were doing. And again, especially with like the high turnover rate and Ophidiomyces Lake, some people don't even know that Cubes is there. And I think just having those opportunities can help strengthen the community and help spread awareness and education as well too. Also, our study is going to go, Greg has 20 years of mark-recapture data on northern map turtles in this lake. So we're going to compare what the mark-recapture data is saying with what folks are saying, and there's going to be that opportunity for collaboration there, which is very exciting. And I just wanted to end it for some hope for the future. These are quotes from interviewers, and these are photos from the interviewees as well that they sent it to me after our interviews of just how much they enjoy watching the northern map turtles on the lake and how important the turtles are to them. So I just wanted to share it with you all because the work that we're doing is important. And I think as a conservation biologist, this gives me motivation, but also as someone who cares about the environment, this also brings me a sense of community. And yeah, I hope it does to you too. Thank you.
So, hello everybody. So today I'm going to try to answer a question I've been asked a lot since I've been starting to work on environmental DNA. Can environmental DNA replace traditional inventory methods? So a lot of people are wondering, are we going to become useless with environmental DNA? So we'll try to answer this question today. So first, I'm going to talk quickly what is environmental DNA. Now more and more folks are aware what it's coming. It becomes more and more used. How we can use eDNA for biodiversity inventories. Usually we use it to detect species, but we can also do inventories with it. And finally, I will compare the two methods for amphibian, vernal pool amphibian inventories. So what is environmental DNA? So that's DNA that is released by an organism in its environment. So it can be hair, scales, feathers, mucus, dead organism. All that will release DNA in the environment. So then we can collect sample of the environment. So that could be water, soil, air, and detect the DNA that is in this environmental sample. So eDNA first was used to detect a specific species directly from environmental samples. We don't need to catch the animal to detect it. So the very first study that was published using eDNA was actually to detect an invasive bullfrog in southwestern France. The bullfrog was introduced in France where there are a problem. And this paper was trying to see, can we detect the species with DNA? Is it more efficient than going out there? And since then, it became a very popular method to detect rare cryptic species that are difficult to detect by other methods, or for early detection of invasive species. So that was to detect a species. Now, how can we use DNA to do biodiversity inventories? So basically, we're going to use a principle of DNA barcoding. So we're going to use a standard sequence in the mitochondrial genome that will be used to recognize a species based on the sequence. So first, we're going to amplify the DNA barcode from the environmental sample. Then we're going to use next generation sequencing to get these sequences, these barcodes. So next generation sequencing made this possible because it can analyze a mixed, complex sample. Before we could only sequence individual samples. Now we can put a lot of things in it and it will detect all the sequences. And finally, we will use a reference database. So here I put the bold barcode of life data system database that hosts all these barcodes from many different species. So all the herbs from Quebec are in the database. So that makes it easy to identify. And we compare our sequences with this database, and that tells us what species it is. So basically, we can just take water, sequence it, and see what DNA sequences we find and say, here are the species that are present. So now let's come to see the big question. Can eDNA replace traditional inventories? So eDNA has a lot of advantages. It's very quick. We just need to go there, collect water, filter it, take it to the lab, and we have the results. We don't need much knowledge about the species. So for herbs, we don't have that many species. It's easy to recognize species, but imagine arthropod, insects, it can be very difficult. You don't have that many specialists. So it can be difficult to identify species from specimen, but from DNA, as long as it's in the database, you will recognize it. So to compare eDNA with traditional methods, so we did both of them in Kenok Nature Reserve. So it's about an hour from here between Ottawa and Montreal. So it's a really big private nature reserve, about 260 square kilometers. Part of it is used for forest cuts for wood, but a big part of it is protected. So we started to work there, and we were interested in vernal pool amphibians. So basically, the original question we were asked by Kenok was, what's the effect of forest cuts on vernal pool biodiversity? That's why we started to work there. So we sampled many pools. So here, I'll just show you the result on 12, where we have all the data from all the methods on these 12 vernal pools, and we did first traditional inventory. So we used several approaches. We did audio recordings. So we put autonomous recorders around the pools, and we recorded 5 minutes every hour from 9 p.m. to 11 p.m. So that makes a lot of files to listen to and to identify species. I didn't do that. It's a student that did that, spends hours with a microphone trying to answer what species in there. Then we put minnow traps in the pool to try to catch some of the amphibians. And we also did, while we were monitoring the minnow trap, we also did some active searching around the pools. So that the three traditional methods we used. And then at the same time, we collected a water sample for eDNA. So that's an example of a vernal pool we studied. So we tried to collect 2 liters of water when it was possible, but very often the filter would clog very quickly. So it's either 2 liters or 5 minutes. After 5 minutes, if we couldn't get to 2 liters, we would stop and note the volume we had. And then we try to see what we detected. So in total, in all 12 sites, we found 12 species of amphibians. So there are only 2 that were recorded in the area, but that were not found, but actually they didn't live in vernal pools, so that made sense. And as you can see, each approach detected 10 species. There's a big overlap between the species that were detected by the methods. There are 2 species that were detected only with eDNA and 2 species that were detected only in the traditional methods. So if we look at that, we say, oh yeah, that's not too bad, a lot of overlap. So we try to look, are there some species that are more often detected with one method or another? So we did this little plot where on the x-axis you have the number of detection with eDNA. On the y-axis, the same thing, but with traditional methods. And there you can start to see that there are differences. So there's one species, a green frog, that was found everywhere with every single method. The green frogs that don't breed in vernal pools, but that basically they are juveniles that use the vernal pools for dispersing, and they're everywhere. So that's the point at the top here that was found everywhere. So the line is the equal if it's found the same number of times in both methods. But there are some species that were detected more with traditional methods. So you have the eastern redback salamander. So that makes sense because it's a species that doesn't go in the water. So it's even surprising that we found it once. So maybe dead animals are drowned and we found the DNA. But we have other species like the spotted salamanders that we found much more with eDNA than with the traditional methods. So we have to say that we went a bit late. It was in May because the sites are not accessible in early spring, so we couldn't get there in April. So basically we probably missed the adults when we went there, and there wasn't enough larvae in the water to detect, but there were larvae in the water, so we could detect it with eDNA. So basically you can see, well, the spring peeper is a good example. Also, we don't catch it very often with eDNA, but with the recording, of course, the spring peeper, you hear it everywhere, even if it's far away from the pool. Actually, you can catch it on the audio recording. So basically, the frogs were more easily detected with traditional methods than the ones that sing. So with recording, it's great. But the species that are more cryptic, newt salamanders, are more difficult to find outside of the big breeding period, and eDNA works great for them. So outside of that, we had another question. We wanted to know if the community we detected in each site was similar. So basically, we sampled the same site with the 2 methods. So for each method, we had a list of species, and we tried to see if we found the same result on the composition, on the species composition. So that's where things become interesting. So that's the question we ask. Are the community detected with the 2 approaches similar? And the answer is not at all. So we found, you can see on the left you have the community detected with eDNA. On the right, the one with traditional method, and you can see there's no overlap whatsoever with the 2 methods. So basically, we can go back now to the question we asked at the start: can eDNA replace traditional inventories? So the answer clearly is no. We find very different results with the 2 methods. Each method will detect a completely different set of species. Well, there's an overlap, but if we look at the community level, we won't find the same species in the same sites with the 2 methods. And what we can take from that, it's important to understand the ecology of the species. As I was showing before, there's no point in taking water to detect the redback salamander. It's not going to be there. So people that know the ecology of the species will always be useful, even if it's to go and get water at the same time. But the other important result is that basically both methods are biased. We cannot have an exhaustive inventory of the amphibian community by using either one or the other. So it could be quite interesting to try to combine the two methods. So another point that we can raise are several, as I said, there are several advantages to eDNA. It's very easy to go there, to take water and to run the analysis. So that's a big advantage compared to listening hours of recording. So we hope there will be quickly software to automatically, as they do with birds. It should be quite easy to train an AI to recognize a species, because it takes a long time to listen to audio files to identify the species, but that works great for frogs, basically. Another thing is that traditional inventory will still be needed to estimate abundance. So right now, there are several studies that try to estimate abundance from eDNA. I'm working on that on the blending turtles. We're not there yet. We'll still need to go there and actually catch the individuals. We also need to catch the individuals to get, information on the demography, if it's young adults, males, females, that we cannot get from DNA. So no, we're not going to be replaced by water automatic sampler. There's still space for field herpetologists to identify and monitor herbs. So thank you very much. I would like to thank all the funders of this study, so KNOC in the first place, that asked us to work on their territory, and also all the people that did the field work. So these were summer students that spent a lot of time collecting data. The audio recording processing was Emily, that is a co-author of this talk. So thank you very much, and I'll be happy to take questions.
Hi, everyone. Thanks for sticking around to the very end. My name is Hana Thompson, and I'm a research assistant with Queen's University and the Canada Biogenome Project, which is not what I'm talking about today. So my regular work is not with turtles, but I've been lucky to hop onto this project with Ginger Elliott and African Lion Safari in collaboration with Dr. Gregory Bolte. And we've been looking at body condition in musk turtles and using in situ data to inform ex situ management. So for anyone who's unfamiliar with ex situ conservation, I know most of us are familiar with in situ conservation or conservation in a species' natural range. Ex situ conservation is just conservation outside of the species' natural range. And ex situ conservation can play many roles in species conservation, including helping to address primary threats in the wild. So for example, using a captive population for conservation-based research, training, and education. Ex situ conservation can also help offset the impact of threats through actions like population reinforcement. So for example, captive breeding and release programs, or demographic manipulation. So head starting programs to reduce egg or juvenile mortality in the wild, or establishing a temporary rescue population for short-term threats like construction in their habitat to hold them in captivity in the short term until they can be released back into the wild. Ex situ populations can also be used as source populations to help restore wild populations to their former range through reintroduction programs, or outside of their natural range for things like ecological replacement or assisted colonization. And finally, ex situ conservation, as sort of a last-ditch effort, can help buy time to extinction by establishing short or long-term populations in captivity while there exist threats to species survival in the wild. And assessing body condition can be an important part of many ex situ programs to ensure that healthy animals are being maintained in captivity, especially for when they will be eventually released back into the wild. So one common way to assess body condition is using body condition indices. These are measurements calculated to estimate body condition or wellness of an individual. and usually relate mass to some measure of body size. So an example in humans would be BMI, body mass index, which is just body mass divided by height squared. And in non-human animals, it's also used. So commonly in fisheries is Fulton's condition factor. Similarly, body mass over length cubed. But body condition can also be assessed using sort of simpler visual assessments. So if you've ever brought your cat or dog into the vet, you've probably seen a chart like this, where you can visually assess the animal to figure out if it's a healthy weight. And similar charts are used for non-domesticated animals as well in zoos, for example, for big cats. Again, the overall goal just to make sure that animals held in captivity are a healthy weight relative to their wild counterparts. And body condition assessments have been done for a variety of turtle species. So in sea turtles, there's 2 body condition indices, again, based on carapace length and mass, as well as this visual assessment scale, which you can see in the diagram for instances when they can't get a hold of the turtles to actually measure them. Desert tortoises have a body condition scoring chart ranging from 1, emaciated, to 9, morbidly obese, again, based on visual assessment with photos developed at the San Diego Zoo. In captive African side-neck turtles, body condition has been assessed by comparing their actual body weight to what their estimated body weight would be based on their carapace length. And for eastern box turtle and Blanding's turtle, CT scans have been used to assess body condition, looking at the distribution of tissue and fat. But there isn't really a simple method for a lot of Canadian turtles, and there is a need for it, especially with regards to ex situ conservation. So for many Canadian turtle species, including all of these species, ex situ management programs like head starting or reintroduction are either recommended or currently underway. And when it comes to things like head starting, reintroduction, or captive breeding, again, it's important that animals that will be released into the wild are set up for success in terms of their body condition. So for releasing underweight animals, they might have trouble surviving the winter, whereas overweight animals might struggle with things like escaping predators or catching food. And so the motivation for this research came from speaking to ex situ managers about musk turtles in particular, as there was some concern about them being overweight compared to wild individuals. But when you only have a captive population to work with, you can't really check. So this is what motivated our research goal to create growth charts for eastern musk turtles. And our hope is that these charts can be used by ex situ managers to track the growth of their captive turtles relative to wild turtles. So we used 5 simple measurements. The only equipment needed is calipers and a scale, so common equipment found in most ex situ facilities. So we measured carapace length, width, and height, and plastron length, and weighed the turtles. And I realize this is a map turtle. I will just be showing data from the musk turtles today. And to create the growth charts, we used data from just under 1,400 wild musk turtles from Lake Ophidiomyces, very generously provided to us by Dr. Gregory Bulte. So we all saw his amazing talk yesterday with the map turtles. So these are the musk turtles from the same lake. And then Ginger and I have received or collected data from 92 captive musk turtles from 3 ex situ facilities, so Scales Nature Park, Science-Stational Snakes, and Little Rays. So this is the growth chart we've created for male musk turtles. So can you see my mouse if I do this? No? Okay, that's too bad. But basically I have mass on the y-axis and then the measurement value for the different measurements along the x-axis in millimeters. And then each color represents a different measurement. So carapace width in green, plastron length in orange, and carapace length in purple. We did originally have carapace height in here as well, but once we started looking at the data, it was a little all over the place, not a great predictor. And we were thinking that might be because if they're chunky, they're going to be chunky this way as well. So it was not a great predictor. So we took that one out. And then I've done a linear regression for each measurement type with the correlation coefficients shown for each one. And then the formulas at the bottom are based on those linear regressions. So the hope is that this chart can be used to compare the actual weight of captive turtles to what would be expected of a turtle that size in the wild. So similar to what was done with the captive side-neck turtles. So to get a sort of range of expected body mass values, we can either just plot the data points right onto the graph and just visually compare where they fall. So points that lie above the line basically are overweight for their size. Points that fall below the line are underweight. Or you can just plug the values right into the formulas at the bottom and again get that range of expected mass values. And so this is the same thing, but for female musk turtles, we wanted to account for any sexual dimorphism, so we did separate male and female. You can see the slopes and intercepts of the regression lines are a little bit different, and the females do get a little bit bigger. So this is just an imaginary turtle to sort of show how the graphs could work. So you see it weighs about 270-ish grams, and then this is where its shell measurements fall. And then based on where they hit the regression line, we can get this range of expected mass from about 200 to 225 grams. So this fake turtle, obviously a little chunky, but let's look at some real data. So this is 5 male musk turtles from ex situ facilities. I'll just look at one at a time so we can see where they fall. And again, there's 3 data points for each individual, 1 for each measurement. So if we look at turtle 3, which is the diamonds here, you can see he weighs 207 grams. And based on the formulas, his expected mass range is about 210 to 230 grams. So he's doing pretty good, if anything, a little bit under what would be expected. And if we look at turtle 5, we can see he weighs 154 grams. And this would be his expected mass range of about 111 to 139. So this one might be a little chunky, maybe worth taking a look at his diet. But so yeah, this is just an example of how this could potentially be used to help with ex situ management of musk turtles. So moving forward, this is very new research. We just sort of started looking at the data, but we wanted to present here because we would love to hear any feedback and are looking for potential collaborators. So we work mainly ex situ, so we are looking for collaborators who work with any of these turtles in situ and might be willing to work with us. Or if anyone works in an ex situ facility and is interested in seeing where their turtles fall, happy to share this chart with anyone. In the future, we do hope to expand this to remaining Canadian turtles, especially those with substantial ex situ populations. So we already have northern map turtle data, again, provided to us by Dr. Gregory Bulte. So that growth chart is in the works. And we will hopefully be getting Blanding's turtle and spotted turtle data soon as well. And finally, in addition to the growth charts, we also are interested in developing an actual body condition index for Canadian turtles. So a little bit more of a specific measurement for each species. So we haven't started doing this yet, but some things we're interested in looking at based on what's been done in other species is that mass over length squared or cubed, mass over carapace length times width, or just looking at the residual distance between individuals and the regression lines. So with that, I'd just like to thank Ginger, who had the original idea for this project, Amy Shabbat at African Lion Safari. Of course, a huge thank you to Dr. Gregory Bulte for all of the amazing data. Thank you to our ex situ collaborators for letting us come in and measure your turtles. And yeah, thank you all very much for listening.